Those moments and those interactions with objects when you're really annoyed and you're like, I'm sure that this could be made better. Like, I'm sure that, has anyone else thought about this and to improve this process in a way? So that's what that's where innovation should sit for me. At the core of a boring afternoon interacting with something that you think it's very silly. Jigsaw puzzles are one of those, do you know this like big pictures that you then break down into tiny little pieces, and you can have boxes of thousands, 2000s, 5000, and this is my favorite pastime, jigsaw puzzles. So trying to bring together again the big picture from tiny little pieces scattered here and there. Just follow the flow and try to imagine what the figure is gonna be, and often you just entirely wrong about what you assume each part is, and then things come together. It's like magic and logic at the same time. It's beautiful.
You're a researcher, and jigsaw puzzles, that sounds like a research problem. How would you compare jigsaw puzzles to your research career?
It's actually, well, now that when I think about it, it's crazy because it's exactly the perfect line of work. I work in single-cell genomics, and single-cell genomics is the art of making sense of a large collections of individual pieces, which are cells, and it's exactly the same process if you think about it. So what we do in this line of work, we take big tissues, we break them down into individual pieces, and then we recollect them back, understanding how the big picture was composed. So it's precisely my line of work.
So basically everyone that loves jigsaw puzzles could do what you do for work, right?
Yeah, yes, absolutely.
Yes.
Yeah,
find your passion. So if you love jigsaw puzzles, don't know what to do in your future, maybe that would be something for you, right? Your career is computational biologist. You're at Helmholtz Institute. What exactly does that mean for you? How does your day look like?
So what normally happens is that I interact with a lot of people from different expertises, and they bring in their questions. So in my line of work, what I do is I try to understand these large collections of data, trying to find patterns using machine learning models, patterns that can suggest something about biological processes or dysregulation of a biological process. So my day-to-day is basically interacting with people from different disciplines, from different biological backgrounds, from mathematical and informatic backgrounds, and figure out what is the right way of realigning the pieces together and figuring what is the big picture. So there's a lot of working with a computer, of course, figuring out, doing a lot of data analysis, but also talking with clinicians and talking with experts in their field to see how different parts can be connected.
In your work, I believe you have a lot of words that you use daily that others have no idea what these means. For example, Pumpypes. Can you explain to me what exactly that is?
It's an interesting thing. As computational biologists, we are tasked with a beautiful thing, which is creating methods and creating tools that others will use. And when you create a tool and you have to create a product, you need to think about a name and how this name will be perceived and used by the others. Pumpypes is a method that I developed, is a pipeline that I developed to automate the analysis of single-cell genomics data. So multimodal single-cell, spatial transcriptomics, these are some buzzwords in my field. And it's often very difficult to decide on a name to use for a tool. So it's quite an interesting part of the process also to think about how to name your product.
You mentioned multimodal, but what exactly do you mean with multimodal here?
Specifically, when we speak about biological data, we know that our tissues are the pieces that compose our body are very different one from another, and they're made of multiple parts. And so in the last, in the history of biology and science, we've been looking at tissues and cells with increased resolution. as we moved along, we realized that we could profile, we could collect multiple readouts from those biological tissues. And so everybody, when now I say DNA, everybody knows what a DNA is, right? The helix, the 4 letters, and so on. But there's so much more material in our tissues. And what we have come to realize in the last few years is that looking at all of these different layers of information together, so multiple modalities, multiple different readouts, actually allows us to gain further understanding of the processes that sustain our life and can, if dysregulated, can generate diseases. So multimodal is the possibility to look at a problem with different readouts, as if you would do, audio and image processing with a normal machine learning model. In biology, we read RNA, DNA, proteins, metabolites. So this all these fancy words.
How huge do you think is the difference between multimodal and unimodal if we compare the performance and also like the possibilities we have with this new way of processing the data?
There's absolutely massive differences, in the same way as if you would, thinking about those adding modalities adds three-dimensionality, if you want, adds dimensionality to the data that you're looking at. And that's real for biology, but that's real also for everyday applications, right? And so whilst the unimodal approaches, looking at one channel of information at the time, can definitely provide a lot of information. If we're missing the other modalities, we are missing a depth into what we're trying to understand. And so multimodal approaches have really revolutionized, in my field, the way in which we understand cellular identities, if you want, and all of the computational biology and medical fields. Having access to multiple channels of information allows you to really increase the resolution into understanding these problems.
So it's like we would be watching a movie to do some research, but then also reading a book about it or visiting a workshop. So we combine different ways of learning about data, but of course that also takes more time. And if we now translate that to multimodal analysis, it takes more time, resources. What does that exactly mean in terms of knowledge also that we need to handle this data?
Yeah. It's a really interesting question, because if you think about individual modalities as analyzed one next to each other, so in a sequential and linear fashion, that absolutely you can imagine this problem taking a lot of time. And this actually has been a way of looking at multimodal data with classical approaches. We first read this channel, and then we read, as you say, right? We see the movie, and then we read the book, and then we listen to the podcast. And instead, thanks to the technologies that we have now, and thanks to AI most prominently, we can access to a way of combining, integrating data that is not, that for us human, actually, it's intuitive, it's obvious. Like, we don't look at the world by only perceiving sound. Our bodies, our brains, are trained automatically, have evolved to process multiple sources of information at the same time. And so what we are doing now is not just linearly looking at individual pieces of information, but understanding the connections between them, connections that happen in time and space at different resolutions. So it's a really interesting, it's a really fun time to be alive, because we now have enough data and we have the tools to integrate this full picture, to do something that us humans do normally, and machines now help us do the same.
Since when are you working in that field? Can you give me a number there?
It's really difficult to decide when multimodality starts, because I had the luxury in my career to focus, to look from the angle of a specific biological disease, and then trying to tackle it with multiple genomic readouts. So it's been now, I would say, the large part of the last 10 years or so. So it's always been multimodal. It's always been, let's look at the genomic configuration, let's look at the RNA readout, and let's look at images. So it's always been multimodal since I started, and I've been privileged to be, to start my scientific career in a world where multiple these technologies now exist.
Right, now a very naive question. So we have ChatGPT, and ChatGPT is multimodal, but why can't you just use ChatGPT for your research?
There's a process that goes into understanding how the results, how the questions make sense with respect to the data. ChatGPT and those technologies are incredibly powerful because they can give you a snapshot of what is going on in the data, but they don't know just yet what is important with respect to what you're asking, how the context of the disease that you're looking at is important, how the answer is perceived with respect to the prioritization of what findings you want to put down first, both in writing a story, writing your paper, writing your grant, but also announcing that to a patient, for example. So these tools are incredibly useful in making sense, reorganizing, creating an order into the vast, disorganized collections of data. But we need humans, we need scientists to still try and connect the dots between the different things and present that to other humans, present the results of the scientific process in a way that other humans can understand it.
How important do you think it is to be able to communicate the value that you bring with the technology that you're building and the research that you're doing, to really enable that research and that innovation to happen?
Absolutely, it's absolutely important. The same way as you build something and then nobody buys it if you don't announce it to the world in the right way. So it's really, really important when you're creating a product to place it, understand who's gonna use this, who am I helping with this, what is the question that I'm trying to solve, whose problem I'm trying to solve. And that's why it's so important to communicate clearly the scientific value of your innovation, the product that you build.
Do you have a framework that you follow there, or are you just trying things out? Like, what is your process to make sure that the communication is on point?
It really depends on the type of problem that you're trying to face. So as a scientist, as a classical academic scientist, I am privileged in the way that there's channels that are there for you. So normally the story is you start from a question, you start from some data, you start from a problem, and then you investigate, try to build a story, and then you write a paper. And then when you're writing your paper, you focus on what is going to be the main message. And that depends a lot on what was the question to start from. But when you're, I don't know, if you're working at the interface between startups and an academic environment, or at the interface between customers, because you're, developing a method, you're developing a protocol, let's say. So it's really important to really keep very, very close what the needs of the person that reached out to you were. And so understanding that, the priority for the client, the priority for the people that are looking at, that are going to use this tool, that are going to use this product or this protocol, I want this to be fast and I want this to be accurate. You really, when you're giving them back the product that you co-developed, if you want, it's important to put the accent on those points and saying, what in the process did I control so that what you wanted is really happening. And that's what makes it, sometimes when you work in this multidisciplinary environment, when you work with this at the interface between different people, you are dealing, it's a privileged position because you're dealing with people that effectively speak different languages. And so if you can communicate with confidence your process, what was the process, and that's why communicating, it's the most important piece, I believe. You can connect people even if they don't speak the same language, and that's really, really important. So keep at the heart, keep close to the heart what the questions were and what was important to solve for you and for who asked for that product.
That brings us to the Human Genome Project. How do you think it was communicated back in April 2003 that made you decide that this is a career you want to pursue?
I think so. I think for the 12-year-old that saw the TV and thought, wow, that's cool. I think it was a really, really good communication, right? Because there was this. I remember seeing the images of, I think it was Tony Blair, right? And then was Bill Gates, and there was Bill Clinton. No, that wasn't Bill Gates. I mean, it was a lot of very famous politicians in the US, in the UK, all over the world, and communicating how important this discovery, the ability to have sequenced the human genome, not entirely, of course, it was finished 20 years later, if you think about it, but how important it was, how groundbreaking it was perceived, and it was such a well-designed campaign that made me decide that that's what I wanted to do when I grew up. I didn't even know what bioinformatics was. I had no idea what a genome was, beyond biology in third class, right? And third grade. And here I am, 20 years later, thinking, yeah, that worked well. So being able to communicate the importance of a scientific discovery, like to connect it to human lives, to connect to daily life, and says, yeah, sure, you discovered the genome, but what is it gonna do for me? I just drive a tram. Like what is it gonna do for me? And just communicate. Look, this opens new doors. This creates a new way in which we understand things and we cure diseases. Yeah, that was definitely well done.
You had a similar moment during COVID where you knew, okay, this is the right career choice for me. But what was different during COVID, like in comparison back then to this visionary human genome project? What was different?
Yeah. There was this sense of urgency and this sense of this is happening around me, and I could just turn my face and say I don't want to. And so at the time, of course, when a big crisis emerged, and I was lucky to be in the right place at the right time. And so things were shutting down. I was in Oxford at the Oxford University at the Wellcome Centre for Human Genetics in the UK, and everything was shutting down, and there was this sense of panic, but also this sense of I want to do something, but I'm only a scientist. Like, I'm not on the front line. I cannot help people. I cannot cure people. I don't know what can be done. And there was someone near me who said, well, there's people on the line. There's doctors in the hospital. And the only thing that I can do is maybe just analyze this data. Maybe just make sense of the patterns. And that's what I'm going to do. And so I felt that was the right thing to do. It really, really changed the way in which I perceive science and the way in which I understand science can be done. So it was a really incredible time because a lot of scientists came together to really try and understand what was going on and try to communicate what was going on to the population. Because sometimes when people are scared, and we can be scared with or without the knowledge, with or without the tools, but when people are scared, they tend to kind of push away what they don't understand, what they don't want to see. Scientists tend to forget that they're citizens at the same time. And so the idea that we really need to communicate, like to coexist with the systems in which we are embedded. We are citizens, we are part of populations. So we need to be really humble when we say, that's the process that we're following, and that's why we trust what we're saying, and respect all the fear that is part of walking into the unknown. That's what we do. That's our job.
So we're walking into the unknown, but I believe that you also have a lot of processes set up that you follow when you start new projects. Can you guide me a little bit through how you start a new research project, if there's a process behind that you follow maybe for yourself or like your whole department?
So normally a good project starts from a good question, and there's an indescribable power in a well-asked question. Normally what happens is that I'm presented with, there's different ways of asking questions. Now some people will be like, okay, the question is, how do I cure cancer? And that can be a question, yes, but I don't know how good it is. And so sometimes the question can also be, I have this incredibly big collection of data and I want to make sense of genetic variation at the single cell level. And so you're tasked with a question that can be biological, can be very technical, can be many things, many layers. And what I do normally is I spend a lot of time listening and studying the problem. So what I would do is, if I'm not familiar with the technology, or I'm not familiar with the biological problem or the disease, I read about it, I ask questions. I'm in service of this question, and so I chase it, and I don't let go. And then I try to understand what are the advantages and the limitations of the technology that was used. Often what happens for me is that I'm presented with data that was generated with one or two technologies, and so I try and understand what is the resolution that I'm allowed, what is the statistical power. If I cannot design the experiment, sometimes it also happens that you're not tasked with the design of the experiment, you get the data afterwards. And then I start breaking down the problem into tiny pieces, pieces that I think would make sense with a bigger story. And you're like, okay, let's validate first question A, and then question B, and then question C. As you enter the problem and as you try and validate your hypothesis, how you think about what is going to be the resolution, how is going to be the narrative that you're gonna say. Also, often happens, it always happens, that what you thought was a linear process actually takes you somewhere else. As frustrating and as scary as it sounds, it's actually the beauty of this job, is how you end up being surprised by the unexpected. So you can plan, and you should plan for technical checks, making sure that the data is sound, testing, using positive controls and negative controls in a way, testing with different, if you've developed a model, for example, you want to test it on different data sets, as many as you can, to validate that your model is going to generalize between different questions. But you also need to be very ready to let the data speak. So you have to have a phase in which you're like, okay, I'm testing this hypothesis, I think that this is the mechanism, but is this what actually the data is telling me? That is very much a creative part. So there's a structure, and there's a let's sit with it. Let's sit with this, what the data is doing, and see if the unexpected is happening.
I know that in a lot of industries we have these gold standards on how to handle things, right? Use this algorithm on that data. If this is the case, use the other algorithm, and so on and so forth. But in your field, it's emerging very fast. So how do you make sure that you're using the latest and the best gold standard, whatever that is out there? Because I feel that's very difficult.
Yeah, so this is an interesting question, because there are emerging a lot of studies that benchmark different models, and I think this is where the field is gonna automate a lot in the next few years. Having the possibility to benchmark, so to design experiment, to actually design and create data, to simulate it in silico, or actually create experiments with real data, where you know what is the answer, where you know what is the perturbation, what the mechanism was, and so what are the numbers that you should see. This is incredibly helpful because it allows you to really see, okay, the model that I'm thinking of using is actually doing what I think it should do. However, one should always remember that using a model in a specific biological, in a new data set that nobody has ever seen before, will probably show results that you don't expect to see. So it's important to have a process, again, to have a process that is sound, to have controls, to have checks that can be as automated as possible. And I think a lot of automation is going to go in this direction, again, as I said, in the next couple of years, on bringing together all these very many methods to tackle dimensionality reduction. And so I want to see that after I've done my dimensionality reduction, the quality of the data is as I was expecting. That you should hit a number, but you should also be ready to understand that what was best at the benchmark may not be working perfectly for you, because your data has other complexities that the data that was used to benchmark did not contain, and that's also totally fine. So the process that goes into deciding which model you will use is not just I'm gonna just use the best, because maybe the best is computationally heavy, and so you don't have the computational resource to apply it, or maybe the best is actually optimizing for a specific metric, and so it's kind of cheating at the game. So you also need to kind of be open to be trying other routes regardless of what the benchmark says.
Let's imagine you have no restrictions regarding resources. You have all computational resources you need, and you can have as much data in one area as you want. What would be a problem that you would want to solve?
I'm quite interested in the way in which we understand the emergence of diseases with respect to life circumstances and the context in which people live. And so if I had this huge grant, this huge, power, what I would do is to collect data, which is biomedical data, genomic data, clinical data, electronic health records for people in their environment, and use machine learning, use AI, use methods that allow you to make sense of this context and say, I see the emergence of this disease in this region of the world, and I think this is connected with what the air quality looks like or what the water quality is. Understanding the context in which the diseases emerge is incredibly powerful because it allows to really see that there's nuances, there's different environments that can generate the emergence of a particular disease, which can be an infectious disease, or can be heart disease or diabetes, and having that piece of information, there's so much interventions that you can then design. So you don't just treat a patient as if it was a human, a citizen, as if it was just a broken system on its own. You understand that it's part of a larger system. And so there's so many other things that you can modulate in order to reestablish a healthy baseline, if you want.
So basically also improving the environment around the people and patients. That is very powerful. So if we now think about the technologies that are currently emerging or we already have, what do you think supports this wish for improving the system around patients? For example, if we think about devices that can track patients, so that we can monitor healthy people collecting their data. Which trends do you think support this wish for creating better environments for people, or also prevention measures?
There's as I said, it's an incredible time to be alive, because now we can collect data from people that are not patients yet. As you said, we can create these huge biobanks and store and profile all these channels of information. But the technological advantage that we now have, that we didn't have 20 years ago, is the possibility to have this distributed systems of analysis and to apply complex, incredibly data-hungry machine learning and AI models. And so what I think is really, really important is the infrastructure that allows you to keep the data safe, keep the data stored at a high quality, ensure that people understand what the data is going to be used for and how it's going to benefit them in return. We are privileged, again, to have witnessed throughout the evolution of my career a moment in which people were arguing whether they wanted to opt in or out of giving their genomic information. And now they're giving away all the readout of the heart rate or the activities that they do, and all the parameters and so on. And there's a lot of responsibility in the way in which we will then use this data. So it's important that both public players as well as private players in this field can innovate with important questions at the heart. Communicating what are going to be the processes, what are we going to do with this data. You are granting us the privilege of having access to a description of what your life looks like in a day. It's an incredible privilege. So communicating clearly to everybody that opts in, say, and we're going to use this data because we want to understand if in 10 years' time you have a risk of developing cancer. If your daughter has a risk of developing cancer, if the quality of air that you're breathing is not the right, is not top, and we want to make sure that we have systems in place to control and check that your life can be the best that you can live.
Maybe I also want to talk about your current projects that you're working on and very passionate about in this context.
During the COVID pandemic, the biggest thing that happened for me was that I was co-opted to be part of this big multidisciplinary group of scientists. We were studying the emergence of COVID and the infectious mechanisms dysregulated when the infection happened. And that really brought me to appreciate how wonderful, how incredibly powerful it is science when it's done in collaborative environments. So I have the luxury now to also be part of this big project, the Cardinal project led by Professor Nicole Soranzo at the Human Technovolume and Welcome Center, the Sanger Institute. In Cambridge. I'm part of this big team who's creating the largest single cell cohort ever analyzed, ever created in one shot from otherwise healthy people, right? People that are not patients yet, that may have been patients for some diseases. And it's patients from the UK Biobank and the Genes and Health cohorts, and so it's multiple ancestries, different medical history, and it's an incredible possibility because I can really see how when you have access to this amount of data at this unprecedented resolution, you can really start seeing changes that otherwise you wouldn't have linked to a specific disease. So you can see the emergence of a specific cell type is linked to the smoking status of particular patients. And so if you can say, if you smoke, this is what is happening to your immune system with such certainty, and say maybe smoking is not good for you. It's something that it really conveys how important it is to create those infrastructures, to create those spaces where we can generate these large amounts of data. They can be safe, they can be looked at in such depth in this collaborative environment. So it's really, as you say, it's really exactly in line with what my passion is, and I'm really lucky to be doing this project.
Now we have these healthy patients, but what does that mean? Can we now detect basically anomalies in other patients and see, okay, they are sick very early, or what exactly is the impact of that?
Yeah, that's exactly what we're trying to understand, right? Because we have the medical history of these patients in certain instances, so we can trace back what their story was, and understanding how this connects to the readout at the single cell level is also important to really break down into tiny pieces what is going to be, whether having or not a specific history gives you a predisposition to manifest a specific phenotype. That's what we call it, a specific trait. And so the possibility to really link together all these multiple modalities in my mind allows you to trace the trajectories of evolution of patients' health. And as you say, it's really important to have access to a lot of healthy cohorts. Otherwise healthy, right? What is healthy then? It's a really difficult question also to establish who of us is really healthy. And so having access to multiple modalities, right? Different readouts. We go back to this idea of dimensionality. Really will allow us to trace trajectories with much increased resolution, with much more accuracy, and hopefully say something about those were the events that probably cascade into the generation and cascade into the emergence of disease.
Now, you worked in a lot of multidisciplinary teams, and it worked very well for you. What was your secret to making these different people work together in a good manner? Because it's complex, we speak different languages.
Yeah. The important thing is to listen, is to be able to sit at those tables and let the fears emerge. Create a safe environment, create a safe space. And safe means we all have questions. We're all walking into the unknown. So we all have things we don't know. And there's things that we don't know that we are aware of. And there's things that we don't know about. What is really powerful there is to create an environment where people feel safe in speaking up about what they don't know. because that's where lies the key to understanding, all right, then we need to go in this direction. We may have the tools, we don't have the tools, so is there anyone in this team who can allow us to build tools to get there? So listening and actively creating environments in which people feel comfortable sharing their ideas, saying, I disagree with what you're saying, I don't think it's a good approach, or I have a different opinion, but I'm not gonna treat you badly just because of that. This has been the key for me, and it's an incredibly enriching experience. It's a humbling experience because it always puts you at the forefront. I mean, at the end of what you know, at the end of your comfortable position in your comfort zone, but you learn a lot. And I'm very, very grateful for having had the chance to do this.
You also supervise a lot of people on your research career so far. How do you approach this? How do you select the people that are worth supervising?
I don't think that there's one rule or a set of rules that you can always apply. What I've noticed though is that enthusiasm is more important than possessing already a technical ability, and nowadays even more. What I see, we have access now to knowledge that is disseminated in every possible channel, in every way. And so finding talent doesn't always mean they have to go to that university and they have to be part of that course. So it's important in this context to be able to listen to the story of the person that is talking. They can have a really good motivation, and of course, it's important, but they have to be curious about something. They have to really want to chase an hypothesis. They really want to give their best to understanding something. And that's how I try. That's how I select the people that work with me. I also nudge them into that direction. I always remind them, look, I have an idea of how this may work, but it may be entirely wrong, because that's how we work. We are right until we're wrong, and proving wrong someone, it's super useful. So I always actually say to the people that I supervise, this is how I think about it, but you need to prove me wrong. And that's absolutely wonderful when it happens.
How do you feel if someone proves that you were wrong? Are you impressed? Are you proud of that person, or insecure? What does that make you feel?
Oh, of course, there's always going to be an element of insecurity, right? There's always going to be a part of you that says, I didn't see that coming. But science is not about me, it's about science. It's about the question that you're solving. And so we collectively are walking into the unknown, discovering pieces. So who do I think I am to think that I know everything about it? And in that way, that becomes also more fun exercise, if you want. You let go of all the anxiety, expectation, you need to perform. Making it into a performative act is also what makes it stiffy and really difficult at times. So also letting go of the idea that you need to know everything, because that's not the job. My job is not to know everything. My job is to guide the new generations and also with them guide myself into learning new things and discovering new things, into being elastic, being agile, into be able to change my mind.
What is, in your opinion, the most important software that you currently use in your team?
I write code, right? So for me, the ability to use a good environment to code is the most important thing. What is really, really important is that you use a software that allows you to write code and allows you to understand what you're doing. Having those plugins with LLMs that allow you to say, okay, this is what I want to do, so that's how the lines look like, but also understanding exactly why this is working in this way. It's incredibly powerful, and I think this is changing the lives of so many researchers, and it's super great.
I want to start in that field. What exactly do I have to know right now to really get started? What do you think is important right now?
I would say that what I would do is, if I were to start right now, what I would do is go and read papers. And I would try and see, the first thing that I would do is, okay, what is fashion right now? What are people looking at and what are people studying? And right now, what is fashion for many reasons? We can talk about hype if you want, but is the possibility to use LLMs in biological data. And so what I would suggest to whomever wants to enter this field is to read about these models. What do they do? What do transformers models do? Is that the best way in which we can analyze biological data and understand, ballpark figures of what exactly is this biological data that we're trying to analyze, and develop opinions about it. They don't have to be right or wrong. They have to understand what are the mechanisms, what are the processes, and ask themselves questions about, is that the only way in which this can be done, or could there be other ways? And this can be, when I think about other ways, I'm thinking about technical ways of solving, the idea of learning context, learning self-supervised patterns from data. That can be technical if you are, someone that enters the computational biology field and come from a very technical background, machine learning, informatics, computer science, or mathematics. But it can also be biological, like someone, or even I've talked to people who had incredibly mixed backgrounds. Someone was doing bioinformatics after having done studies of language, so how languages evolve and why they have that structure. And so if you come from the more applied, if you want, fields of how biology works, ask yourself, is this the right way of understanding, of learning context from biological data? Is that how a cell really works, putting things in order, and the order makes sense, the order of expression of the genes makes sense? So ask yourself the questions about what you see are the big papers coming out, and that is gonna guide you to think about, again, a process that you are annoyed with, something that you think is not really working well, and that's gonna generate your hypothesis that you want to chase.
Can you put a number of the dataset sizes you have and sample sizes you have?
Yeah, so it really varies with respect to the project, but as I was saying, right now in the single-cell genomics field, we're working with atlases. So as part of the Human Cell Atlas, whose mission is to collect, to profile all the cells of all human tissues to gain a comprehensive map of complex organisms, such as the human body. We are working with the order of millions of cells. We're almost at the billion level, almost, I guess, if we put everything together. And right now, interestingly enough, as I said, finally we are working with large-scale cohort level. So we're talking about thousands of individuals. But this thing is just only doomed to increase. So the order of magnitude from the first experiments in single-cell genomics has increased quite a bit. So we went very, very fast from one patient, a lot of cells, to thousands of patients, millions of cells. And that in data is terabytes and terabytes and petabytes actually of data. So it's actually the complexities involved do not just it's not just making sense of the patterns. It's also, do I actually have the software that allows me to access the data fast and analyze it and produce the results, reproducibility about those processes? Or like, you need to really have systems in place, to infrastructure in place to accompany, like to guide the data from the generation to finding the results.
If you train your own model, how long does it take to train?
Depending on many variables, right? What are you trying to answer? But it can take anywhere between a couple of hours to 2 days or a week, depending on the size of data, depending on what you're trying to do. So if you're training, for example, a single cell foundation model, which are very famous these days, if the code is very well optimized, it can take days. Yeah, let's say days.
Now, if you look at code development, how much, like percentage-wise, of the time do you spend on optimizing the speeds the processing speed for your algorithm?
Well, there are phases in the development of a model. And at the beginning, it's actually absolutely fine that you just prototype something that is kind of gonna work. And so you have to have, so in the first phase of prototyping, you have to have an idea of what you want to achieve. And you just write, the pieces as if they were Lego blocks, and you just write what works. You test that the hypothesis works, and then you go into a pre-production phase in which you need to optimize the speed and the efficiency, the computing efficiency of your algorithm. So I feel in the academic environment, we are limited to the extent to how much we can spend on optimizing models for speed and for efficiency of computational resources. But this is becoming more and more important and more valued. If you can build a method that will work out of the box and be really, really fast, then it's going to increase the chances of uptake by the general community. Since a lot of people from computer science and from the more technical fields are coming into the computational biology, I feel the quality of the method is increasing for everybody because the checks are increasing. But we're not there yet. We are not at the place where things are perfect out of the box and are efficient out of the box. So we do spend time doing specifically that, making models as efficient as they can. But you can always improve on that.
If we now look into your team size, how many people work on such a huge project, approximately?
It really depends on the project, right? So when you're working with these large projects, the teams are large. But normally you get 10 to 20 researchers all involved on all having some level of technical involvement in the analysis, and it's a challenge in itself because we don't come from the same we don't speak the same language, we don't come from the same training, we work online mostly. It's also another challenge to interact with people that you've never seen in person. So it's a small village.
So you're basically raising a kid there. Can you tell me what would be like the one sentence, maybe also rule that you set up for yourself throughout your career that helped you to really stick to learning, having that enthusiasm, believing in what you do? Is that the impact that you saw? Or is that really some mindset that you have as well?
There's one guiding principle in my career, and that has always been curiosity. I was always very curious about what was going on in a field, in a particular disease, in a new technology, in a new protocol or a new machine learning method. So that for me has been a guiding, really a guiding principle, a guiding light. And of course, at different stages of your career, you will have different motivations, and that's absolutely fine. The thing that really humbles me and really fills my heart with warmth, if you want, is when I speak to the students that applied, to do a PhD, and everyone with no exception, I think it's beautiful. Everyone say, I want to do a PhD in computational biology in general because I want to help people, because I want to find a cure for a disease that they are familiar with for some sad reasons. And I find this beautiful. I find this really, gives me hope in humanity, if you want. And that is an incredible, it's an incredible incentive, right? Wanting to do something for others. But it also have to come from for me, I need to be curious about it. I need to really want to understand something. And so there's no way in which you can be curious about something if you don't allow yourself to not know something. So it kind of goes back to the question that you were asking before. What do you do when you realize that you've been proven wrong? There's a moment of, oh my, and then, oh no, that's exactly what was supposed to happen. So I don't know about something, that's why I'm curious about something, and that's my guiding principle.
I feel like this is a very beautiful wrap up for our session today. Thank you so much for being here. I've really enjoyed our conversation. Thank you.
Thank you, Julia.